In this video, we are going to be moving away from mitosis and into meiosis. So meiosis, unlike mitosis where we are just dividing our body cells, we are going to start talking about reproduction and how we form sperm and egg so that we can actually combine to form a viable offspring. So our focus in this unit is specifically reproduction. Reproductive cells, how do they get created, how do they combine to form viable offspring, and how can, as we move into genetics, how can we see diversity and different variations start to appear in these offspring. But first we have to review. So in mitosis, we had talked about the difference between somatic and gamete cells. Somatic cells are your body cells. They are diploid, so your muscles, your skin, your nerves, all of those are forms of somatic cells. They're going to reproduce through mitosis, and they're always going to be diploid. They're always going to have two copies of their chromosomes. We are moving then into gametes. Gametes are our sex cells, our sperm and our egg. We could also call them germ cells. They're going to be haploid, meaning that they only have half of their chromosomes at one time. For example, in order for a human to form, we need to get half from mom, half from dad. So that's where this haploid comes in. And these are going to reproduce through meiosis, which we're going to talk about. We also, going into, again, diploid, same amount of genetic material as the parent. So in humans, we have 46 chromosomes. So all of our somatic cells will have 46 chromosomes. Germ cells are going to be haploid, meaning they have half of the genetic material. So from a diploid individual, meiosis is going to produce haploid individuals with 23 chromosomes. So this process is going to be very similar to the process of mitosis. The big thing, though, is we are going to end up going through all of these stages twice. So let's start out with chromosomes. We focused a lot in mitosis about chromosomes. We're going to focus a lot on them again here. So take a moment, and I want you to make as many observations as you can about this diagram. Okay, so you may have noticed in this diagram, this is a karyotype. So this shows all of the chromosomes available in a human. You notice that all of them are paired up, and they usually have about the same size, the same shape, the same location of their centromere, except for this X and Y group down here. So this is a human male, and these pairs of chromosomes are called homologous pairs, otherwise known as tetrads. So homologous means that they are the same structure, the same shape. So for example, they are the same height, their arms are all the same length, as well as their centromere, this section that holds the chromosome together, is in about the same location. So you can see in chromosome 1, our centromere is right there, our arms are all about the same length, and we overall have the same length in our total chromatids. So at the beginning of meiosis, we want to create homologous pairs. And we're going to see why this is as we move through meiosis. But like I said, these are called tetrads. Tetra means 4. If you've ever played Tetris, all of the blocks in Tetris have 4 little subunits because tetra means 4. So in this case, we have 4 chromatids. 1, 2, 3, 4, which is why it's called a tetrad. And these tetrads are going to go through a process called crossing over. This is something we don't see in mitosis. This is where we get the swapping of genes between homologous chromosomes. So a couple stages. We start out with synapsis, when our chromosomes actually connect together. They kind of wind around one another. Crossing over itself is the process of swapping their genes. So on chromosomes, or homologous chromosomes, each of the arms should have the same genes in the same locations. And so by crossing over, we can swap these sections to give the other half to, or give the other section to the other chromosome. And this allows for a lot of genetic variation, which we'll see a lot more when we get into genetics. And finally, we get to disjunction, where we can see we have these recombined sections in our new chromosomes. And these chromosomes are going to follow something called the law of segregation. So this also plays into genetic variation. And the law states that each gamete is going to end up with one copy of each gene. So we want to make sure that when we are doing crossing over, each gene successfully gets transferred over. We'll talk about what happens when that doesn't happen. But each gene needs to be on each chromosome so that when we get our possible gametes, we can have each specific gene. We can make sure that all of the instructions are there to code for our new organism. So this is an activity we did in class. So for those of you who are out, you can come see me and I can get you set up with this. But we are going to move now into the actual stages of mitosis. And in meiosis, there are 8 to 10 stages, because we go through PMAT twice. So if you remember from mitosis, our acronym PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis at the end, is going to happen twice. So we start in prophase 1. In prophase 1, we get some of the normal characteristics of prophase. The chromosomes are going to condense into the chromatin is going to condense into chromosomes, the nuclear membrane is going to break down, the spindle fibers start to form, but we form tetrads. Normally in mitosis, we just have our chromosomes condense and sit around. In this, we are going to form our tetrads and actually do crossing over. So while the nuclear membrane is breaking down, while everything is getting ready to divide, we are going to swap genes. Once this has happened, we move into metaphase. Metaphase, typical characteristics, the chromosomes are going to line up at the metaphase plate in the equator, so this middle band around the cell. In this case, though, instead of the individual chromosomes lining up, the homologous pairs are going to line up at the chromosome or at the equator. This is going to be followed by anaphase 1. In anaphase 1, very similarly to what we saw in mitosis, the chromosomes are being pulled apart by the centrosome. So you can see the spindle fibers here are connected to each of our chromosomes. But instead of getting separated like we did in mitosis, in meiosis we are pulling the whole chromosome away from its homologous pair. And we call this process, not only in meiosis but also in mitosis, we call this process disjunction. So the spindle fibers pull all of the homologous pairs apart, and we move into telophase. At this point, our chromosomes are at each of the poles being the opposite ends of the cell. They're going to start turning into nuclei, and the cytoplasm is going to divide. We can see here during telophase, we have our cleavage furrow, the point where the cell kind of pulls and pinches apart. And we are left with two cells that are haploid. Now, this seems a little odd since in mitosis we made two cells that were diploid. And the reason is because we have half of our total chromosomes. So let's say, for example, the red ones originally came from mom, and the blue ones originally came from dad. Now, in reality, these could be any which way. There's no real... these all came from the same individual, so it would have to have came from much farther up. but for the sake of explanation, the blue one is dad and the red one is mom. And so our original cell had 3 from dad and 3 from mom. And so when we performed meiosis 1, we separated them in half. So only one of each gene from dad got sent to one of this daughter cells, and only one gene from mom got sent to the daughter cell. And so this is why they are haploid. Even though we still have our chromosomes together, compared to in mitosis we had individual chromatids, in this we are separating, so now there's only one version of each gene in each of our daughter cells. So take a moment and kind of think about how this was different from mitosis. So there are a few differences between meiosis 1 and mitosis. For example, in meiosis 1, we start with our tetrads, our homologous pairs, while in mitosis, we only start out with individual chromosomes. In mitosis, we also don't perform crossing over because we aren't looking for that genetic variability like we are in meiosis. Finally, in meiosis, our daughter cells, the ones that we come out with at the end of meiosis 1, are haploid instead of diploid, which is different from what we saw in mitosis when we came out from having diploid cells. So now we move into meiosis 2, and this happens very, very quickly after meiosis 1. So in prophase 1, again, we are setting up like we did in mitosis. Our nuclear membrane is dissolving, our chromosomes are winding up and turning into chromosomes, and our spindle fibers are forming and getting ready to pull apart our chromosomes. This is followed by metaphase 2, when our chromosomes again hook up to our spindle fibers and connect in the center of the cell along the equator. And this is going to be very similar now to mitosis. In anaphase 1 or anaphase 2, we are dividing our chromosomes into their individual chromatids. And they're going to move towards the poles, as we've seen before. And finally, telophase 2 and cytokinesis are going to split our cells into 4 new daughter cells. And again, all of these cells are haploid at this point. So for example, in these two up here, we have one copy from each parent, or we have 2 copies in each cell. Same thing down here, we have these 2 copies in these 2 cells. So all of our cells now have 23 chromosomes, just in the form of chromatids instead of homologous chromatids. So from this, you should be able to go through your poster, try to draw and label the steps of meiosis so that we are able to better compare it to mitosis. If you want more practice, there is also a meiosis square dance that kind of goes through. Overall, the process of meiosis is very similar to square dancing if you look at how the cells divide and everything. So this is just kind of a fun way to remember the stages of meiosis. So this is where this video is going to end. Over the next couple days, we're going to continue discussing similarities and differences and kind of compare meiosis versus mitosis. We're also going to jump into reproduction, which will be in another video, as well as diseases that can arise from both mitosis and meiosis. So if you have any questions, feel free to email me, and I hope to see you in class.